Good morning. This is YouTube video AP Bio Review 18, and it's covering experimental design. The reason that we're looking at experimental design is because question number 1 this year, you might not be able to see this very well. That's okay, I guess I can zoom in on it. Let me zoom a little bit. Question 1 is supposed to be worth 65% of our grade. Again, this year we have 2 free responses. And question 2 is supposed to be 35% of our grade. So I know it's not exact, but that's basically 2 to 1, 2/3 of your grade and 1/3 of your grade. Now, if you look at this, question 1 presents students with an authentic scenario accompanied by data in a table or a graph. That means lab. They're gonna tell you about some enzyme or some gene that you've likely never heard of and kind of tell you what it does, give you some data, maybe some graphs, something like that, some drawings and such, and then start asking you questions about it. So we're kind of thinking it's going to have an A, B, C, D, and it's worth, you can kind of think of it as like 8 points. This over here, A, B, C, D, and it's kind of worth 4 points. Again, not exactly, it's 65-35, but more or less. So part 1, describe and explain, or part A, describe and explain biological concepts, processes, or models in applied context. So you're gonna have to be describing what's going on. That's kind of vague. Identify and justify experimental design procedures. So what's the significance of group A or B, or what's a control? Who's the independent variable? Who's the dependent variable? Here's a graph. What's the dependent variable? That type of thing. Analyze data. That's exactly what it sounds like. You'll have to look and sort of, after looking at the data or the graph, determine like, well, what the heck does that mean? Does this drug work, or is this gene being turned on or off, or is this enzyme denaturing, that type of thing? And then make and justify predictions. So now they might say, hey, what would happen if, let's say we're doing an enzyme experiment, hey, what would happen if we increase the temperature further? And you might say, well, the reaction rate would go up a little bit because substrates and enzymes collide more often, but if it gets too high, then the protein would denature, and therefore my prediction is the amount of enzyme activity would go down or something like that. So this part right here is going to be very experiment heavy. Over here on part 2, there's also room for experimentation as well. Describe biological concepts or processes in applied context. That's a little bit like the free response that we just did, where on protein synthesis, where you had like a TRPT or something like that, there was like an enzyme, then it turned into like, it was converted into I3PA, and then indole acid IAA, and there was like a gene mRNA, sort of like that. Explain biological concepts or processes in an applied context, again, sort of like that question. Predict the cause or effects of a change in a biological system. Support or justify a claim with reasoning or evidence. So I can pull that up real quick, the one that I just referenced. So bear with me for just a second. So for question 2, we're going to need a shorter version, kind of more like a 4-point version, compared to this one that I'm getting ready to show you. But I think that this is sort of what they mean for like question 2. This question that we were looking at, 2019 number 1. Now back in 2019, this was a 10-point question because it's number 1. So we'd have to bring it down to, about a 4-point question. But I think this is what they're talking about in 2. All right, so what we want to do now, let me adjust the camera, is go back and walk through experimental design. You've learned it many times, but it's guaranteed to be on there. And one of the bad things about learning it many times is you just assume that you're good at it, but sometimes students kind of still have some blind spots. So these are the things I want to make sure that we cover. I don't really have any free response to reference up here on the board. Down in the description, I'll probably put a few. So make sure you kind of look down in the description of this YouTube video, and I'm sure I'll put a couple of them in there. Okay, so first off, constants. Things we always think about in a controlled experiment. If they were to say, design a controlled experiment, they're not. For this year, but if they did, I always think of these 4 things first: controls, constants, independent variables, and dependent variables. So we'll start with constants. Let's say that I want to do an experiment and I want to test, we'll just call this brand green, disinfecting wipes. We'll just call this brand yellow. And I want to see which one is better at cleaning the carts at Kroger. Let's say that SARS-CoV-2, CoV-2 was, instead of being a virus, it was a bacterium. And so maybe wiping down the Kroger carts with this would destroy them. You might say, well, Mr. Cotton, we are wiping them down with stuff to try to destroy the viruses. Why don't you just assume it's a virus? I want to use an experiment with a bacterium because we've worked with bacteria. So we've, in our science class, we've gotten petri dishes and grown bacteria, some E. coli bacteria on there. So you're more familiar with the process. We never cultured viruses in live cells and stuff like that. So we're going to pretend that this whole COVID-19 problem is bacteria-based instead of viral. Okay, so what I want to do is I basically want to figure out which one of these is better for me. Let's say that my job is I'm taking care of the carts. So I bring the carts in and I'm supposed to clean them off for our customers. And I want to do an experiment and see which one of these is better for cleaning the handles. And what I need to do is I want to keep everything the same. So when I'm testing these and when I'm testing these, I want to use the same type of carts. I want to make sure the plastic on them is like brand new and clean, or they're both equally crappy with a bunch of grooves in it. I want to make sure that I use the same type of motion to clean the cart. So let's say that this is a rail on a cart. I'm going to take one of these wipes maybe, and if I decide this is how I'm going to clean it, it'll be like 1, 2, 3, 4. And then whenever I clean the next one, I'll get a fresh wipe. 1, 2, 3, 4. Everything is remaining constant. When I clean another one, 1, 2, 3, 4. So I'm using one wipe from each. I'm making sure they're both like equally liquidy, and I want to use the same type of carts, the same type of motion. I want to keep everything constant, except for one thing, my independent variable. These will be my experimental groups. My experimental group will be based off of my independent variable. So my independent variable is the thing that I change, or the thing that I manipulate, or you can kind of think of it as it's like the reason I'm doing the test. So the only thing that's going to be different is which wipe do you get? Do you get brand green, or do you get brand yellow? Everything else should be the same. Now, my dependent variable, it depends on the independent variable. What does that mean? My dependent variable is how I'm going to measure success, like basically who won the competition. How am I going to measure that? So what I might do is from my dependent variable, I might come back after 1 hour and get a swab. I'll get a clean, sterile swab. 1, 2, 3, 4. And then I got my petri dish right here. And these are actually just like, drink cup tops. And I'll clamshell 1, 2, 3, 4, 5. Or I guess I'll just keep consistent with the 4. 1, 2, 3, 4. Cap it. Then I get another one. 1, 2, 3, 4. Pop this open. 1, 2, 3, 4. Cap it. So my dependent variable is what am I going to actually measure? Am I looking to see which one grows the least amount of bacteria, or like diversity of bacteria, right? For example, if after I grow my bacteria, we might find that this one is covered in E. coli, like 100 colonies. Maybe this one only has 1 colony of E. coli, but it's also got 1 colony of strep. This one has more like total bacteria, but this one has more bacteria diversity. So what am I testing for? You want to be clear. You don't want to just say, for my dependent variable, bacterial growth. You need to tell me how you're going to measure it. Bacterial growth, as measured by number of colonies grown on a petri dish. Or you could even look at like percent of the field of the petri dish or something like that. You need to tell me like what exactly you're going to measure with your dependent variable. So next up, let's say that I do this experiment. So I bring this in, 1, 2, 3, 4. I get another one, 1, 2, 3, 4. I get a wipe and a wipe. I streak my plates. Now I've got to incubate them. Maybe I'll say I incubate them for 24 hours. So I get out my incubator. This is a rare incubator. People don't really know much about it. It's manufactured by Capri Sun. So I'll put those in there. And human body temperature is 37. So I need to set my incubator at 37. So let me do that. 37 Celsius, by the way. If you're 37 Fahrenheit, you're very dead. So let me dial that in real quick. There you go. We'll come back in 24 hours and we're going to check. And according to my dependent variable, I decide I'm going to count just total number of colonies. Okay, great. And so let's say after that, I have type green, brand green versus brand yellow. And let's say that brand green grows 20 colonies and brand yellow 10 colonies. Great. At first we'd say brand yellow is better, but I'm missing something in my experiment. I need controls. So a control is generally what you think of when you think of a control, you're thinking of a negative control. So when you learn the word control in 9th grade, 7th grade, whatever, back in the past when you're doing experimental design, you were always talking about a negative control. A negative control means you don't get the treatment. You don't get this independent variable. So you don't get one of these. So what we'll do is we'll get some sort of cloth. Hopefully it's a similar type of cloth to these, and it won't have any chemical on it, any of these active ingredients, whatever the active ingredients are. We'll just soak it in water, and we'll try to make it about the same wetness, and we'll do the same thing. 1, 2, 3, 4. And let it sit, swab it, bacteria, plate, come back. We might find that the negative control, which was just water, maybe also had 10 colonies. We might find out that it had even less. And so what does that mean? That means that these guys actually suck at preventing the growth of bacteria. So if I've got 10 colonies without, basically it's like, all right, the only thing that I'm doing, maybe it's the wiping motion that's actually kind of just raking off the bacteria. Maybe it's the friction creating heat as we wipe. Who knows? So that's why I need a negative control. Now, what if though I got this? What if I got 0 growth on my bacteria plates with brand green? You'd be like, oh, awesome, this is great at killing bacteria. And I got 0 growth from brand yellow, and I'm like, oh, they're both equally good. But then I also got 0 growth from a towel with water. Well, again, there's a couple possibilities. One, it could mean that, just using anything and wiping along there, or using any kind of cloth and wiping along there is good enough to kill the bacteria. Or it could mean that maybe when I poured my agar plates, I screwed up. Maybe I have some antibiotic in there. Remember how we had antibiotic on purpose in some of our plates, ampicillin? So maybe I've got antibiotic in there, and so basically I just have bad plates. So how would I know? Then I want a positive control. Positive control is where you guys are less likely to be familiar. A positive control would be something that you know has bacteria. So I might get a plate that actually grows E. coli. I can literally see the E. coli on there. I'm going to go in and I'm going to swab some of it. I can see it on here. And then I'm going to swab one of my clean plates, incubate it, just like the other ones, and see if it grows. If it comes up and no bacteria grow, now the reason that these are zeros is because my plates are suspect. So I need to kind of investigate the experiment. If this one right here grows great, if it's got like hundreds of colonies, then that means, hey, that, and that, they really are 0, we think. So that's what we call a positive control. A negative control is where you're basically not getting the treatment. A positive control is one that, based off of past experience, you know should give you an outcome. So that's a positive control. Now, let's say I do this with one Petri dish plate, one of these, and one of these. What's wrong with that experiment? My reliability and my statistical power is going to be low. So I want to repeat or have large sample size to begin with. If I can only do 1, 1, 1, 1, cool, that's fine, but repeat the experiment 50 times, 100 times, it all depends. When you get results that are really, really tight, then you have to repeat more and more and more and more and more. When you get results like 20 colonies, 2 colonies, 100 colonies, and it's like 20 and 2, 21 and 1, 30, and 3. When yellow consistently beats the crap out of green, you might not have to do 100 trials. 50 trials might be good enough. But when they're kind of more neck and neck, you need to do more trials. My point is this. I either need to have a large sample size to begin with, 100, 100, 100, 100, or I need to just keep repeating the experiment until I have hundreds of data points. That will increase my reliability and increase my statistical power. The null hypothesis. Technically, it's fair game that chi-square could be on this test, but if you've got, 40 minutes of actual test taking time, right? You get 25 minutes for the first question, 5 minutes to upload, 15 minutes for the next question, 5 minutes to upload. So you really only have 40 minutes of actual testing time and then 10 minutes of upload time. If they're going to have you working for 40 minutes, I don't think they're going to give you a full-on chi-square problem, because that would be too big of a proportion of your grade relying on just statistical analysis chi-square. But they might ask you for a decent null hypothesis. So a null hypothesis is when you just assume that there is no difference. So you assume that, hey, using water, using this makes no difference. So you're essentially going to think that, hey, putting this on there and putting this on there doesn't kill bacteria. If I was testing my athletes, I know I use this example all the time. If I was testing my athletes to see whether Gatorade or Powerade is better, and my negative control is water, my null hypothesis would be like, hey, however fast my sprinters run, let's say that that's my dependent variable, their time in the 50 or something, I'm going to think that there's going to be no difference between water and Gatorade and Powerade. Here, I'm going to think there's no difference between just regular water and brand yellow and brand green. That's my null hypothesis. Now, let's say that there's a difference. Let's say that this one has some bacteria on it, and the ones treated with this have some bacteria, and the ones with these have bacteria, but these guys have less than this. How will we know, is it good enough? So, for example, let's say that my negative control, we go back and put some different numbers up here. Let's say that my negative control had 30 colonies, and the yellow had 25, and the green had 23. All right, yeah, that's different, but statistically, is that different enough? So we want to run some sort of statistical analysis. So again, on this test, you might just say we'd run statistical analysis. If chi-square is appropriate, that's one of the major ones that we use. If you have an expected and you can run a test and see how close were you to your expected, that might be an appropriate test. You could even say I could do a chi-square analysis to see if the differences are statistically significant or if it's probably just due to chance. Those differences might be small enough, like for example this one, those are small enough that even without running chi-square, I'm pretty sure that those are different enough, or that those are close enough to be statistically insignificant. Next up, we'll look at standard error of the mean. Mean is, that right there is our symbol for mean. So standard error of the mean. I have seen tests where they actually write SEM, but that was when they first started introducing biology to standard error of the mean. Now they talk about it like they do in stats. So standard error of the mean and overlap. Let me kind of show you what that looks like. So on your test, it's likely that you're going to see some data tables or graphs, even though there's only 2 questions. And a lot of times they're going to ask you to explain what's happening in the graph. So describe what's happening and then explain why that is happening. So we're going to walk through a few examples of that real quick. There we go. That'll be all right. Okay, so on this one, the graph depicts relative activity of a particular enzyme as pH changes. Describe the shift. So if it ever says describe the graph, you're just putting into words what the graph shows. So describe the shift in relative activity between pH 8 and pH 10. From 8 to 10, the way you describe that is it goes down. So the amount of relative activity decreases. If you had numbers over here to where you could maybe calculate a slope, you could say, all right, it's got a negative slope of whatever it is, rise over run. I don't have any numbers on this side, so I don't know for sure. If they were if they were to say, describe the graph at right, I don't think that they will. But the way you can know if you're describing it correctly is if somebody was not looking at the graph and they were just listening to your description and trying to draw the graph, would they? So what I would say if I was describing this entire graph is I'd say from a pH of around, if that's 4, that's maybe 3.5. So pH 4, we'll call that edge 4. So at a pH 4, the relative activity is, close to 0. It increases up until it hits a pH of about 7. And it has a pretty constant rate maybe. So it increases at a constant rate until a pH of 7. And once it hits a pH of about 7, it decreases at a constant rate. And it's now going in the negative direction until it zeroes out at about whatever you want to call that, 9.5. And so if you just gave me those words and gave me a graph, I'd be like, all right, I increase up to pH 7, and then I decrease down to about pH 10. Cool, and I draw it. Explain why. All right, why did this happen? Well, if it's an enzyme, looks like the ideal pH is somewhere around 7, and it looks like after that, if you go below that or above it, it risks denaturing, and it looks like it's totally denatured by the time you get to like 9.5, 9.7 pH, whatever that is. So describe a graph is what is it doing? Explain is then like tell me biology. If they want to throw something math at you, something that's pretty quick and they do it often is to calculate the rate of something. So rate is anytime you have a change in y over a change in x. Rate is usually time, distance over time, velocity over time, that type of thing. So like distance over time is velocity, velocity over time is acceleration. So it's just rise over run. So they might say, calculate the rate at 5 degrees Celsius. 5 degrees Celsius is the circles. As the temperature increases from 10 to 20, so what I do is I would just go circles from 10 to 20. So rise, it looks like I'm going 120 minus 80, and then the run is 20 minus 10. So I've got 40, a difference of 40 here, over 10, so my rate is about 4. I'm going to need units. This is free response, so I need units. Well, my units are rise over run. So whatever my units for y are, I just put that over this, even if I don't get it, even if I don't understand it. So I might not be able to conceptually visualize what is milligrams of oxygen per gram, per hour, per degree Celsius, but it doesn't matter. Just take that y-axis unit and put it in a fraction over the x, and those are your units, even if they seem really bizarre. Skip over that one. Okay. So now we're getting into the thing that I was wanting to bring in next, standard error of the mean. These little error bars. If the error bars overlap, then that means the difference between these, so like right here, this bar graph right here, the white one versus the gray one, these error bars overlap. So this area in here overlaps with this area over here. So that and that are statistically not significantly different. So whatever happened at hour 0, with exercise, and at hour 1, whatever's going on in this experiment, the difference between those is not different enough for us to say, yeah, they're different. Same thing here without exercise. Those error bars overlap. So whatever this data is showing in this one, it's not statistically different. Kind of like if I said, here's a guy that works out, regular diet, here's a guy that works out, and he drinks our protein powder. And if the error bars overlap, we'd say, yeah, the protein powder didn't help him, whatever our dependent variable was. Maybe his squat max or something like that. His squat max didn't significantly increase. In this one, now you'll see there are places where the error bars don't overlap. So if I look at this data set right here, here's my error bars, here are these error bars. They don't overlap. The lowest guy there and the highest guy there don't overlap. So that data and that data are statistically significant. We really do think that this group and this group perform differently. Now, group 2 and group 3, those overlap. So we don't think that they're very different. 2 and 4, that guy overlaps with that guy. So we don't think that they're different. There, I can't quite decide if they overlap or not. There, we can argue about that, but I think you get the idea. One last thing, they might give you a data table and ask you whether or not they overlap. So let me show you that. So Here's the data table that's on the screen. I know you can't read it right now, so I just wrote it this way. So this is just some data for some experiment at temperature 25, 30, 35, and so forth. And we were measuring carbon fixation. So this is probably an experiment on photosynthesis. It says plus or minus 2 standard errors of the mean, and then they give me units. Okay, so 91 plus or minus 9. What I want to do is I want to see, they might ask, is the difference between temperature 25 and 40, is it a legit difference or not? Is it statistically significant? And what I have to do is just sort of like write out the range and see if the ranges overlap. I was looking at graphs to see if the ranges overlap. Now I'm looking at a data table. So here, 91 plus 9 would mean my range is 100. 91 minus 9 would be 82. So my range is between 100 and 82. Here, 96 plus 5, 101. 96 minus 5, 91. 93 plus 8, 101 minus 8, 85. 104 plus 7 is 111. 104 minus 7 is 97. 78. 71 minus 7 is 84. I'm sorry, 64. Went the wrong way. 53. Man, 63. Keep getting off by tens. Okay, so now I want to see if their ranges overlap. So for example, if I was comparing these two, this guy goes as low as 82, this guy goes as high as 101. So their ranges overlap. Their ranges overlaps. Now, when I start getting down to this guy, the peak of his range is 78. That is lower than their low end. So these four guys all overlap with each other. So there's not a statistically significant difference between the results from temperature 25, 30, 35, and 40. But when we get to temperature 45, when it warms up that much, this is Celsius, by the way, degrees Celsius, then now the difference is significant because that 78 doesn't reach as high as that 97, that 85, and so forth. Then right here, we've got another data point that's significant. The high end of this range is 63. The low end of this one is just 64. So these guys are a group. That guy's his own group, so to speak, and that guy's his own group, so to speak. So it does seem like once you start getting somewhere above 40 to 45, 45 for sure, but somewhere between 40 and 45, it seems like there's a statistically significant difference and my carbon fixation amount starts to go down statistically significantly. Now, basically, if I took these same points right here and drew them on a graph, then you could see that the bars don't overlap. So like, here's how I would draw it. Let me put this up here so you can see the numbers. So I would have 59 plus 4, 59 minus 4. So plus 4 goes up to like 63. So it'd be like right there, 63, and it goes down to maybe 55. So what we'd see is, let me bring that guy down slightly. What we'd see visually is that guy and that guy don't overlap. Visually it might be tough to see, but based on numbers I know that they don't. Same thing here. This guy gets as low as like, 82 or something like that. That guy gets as high as like, I don't even remember what it was, 78. So they don't overlap either. So that's what it would look like on a graph. So I can also see if there's overlap just in data tables. All right. I have seen practice problems where they give you a graph and they just ask you what's the dependent variable or what's the independent variable. So back over here, if I asked you what's the dependent variable or the independent variable, dry mix. You might have heard this before. The dependent variable is on the y-axis. We also used to call it the responding variable, that's why the R. And the independent variable is on the x-axis. We used to call it the manipulated. So all you care about is D and Y and I and X. So my independent variable is on the x-axis. So if they ask, what is my dependent variable, you would say blood plasma prolactin and millimoles per liter. Another thing that you'll see a lot is they'll say, based on the data, describe the effect of X on Y. So based on the data, explain the effect of exercise on whatever's going on right here. Well, it looks like all my data for with exercise and all my data without exercise are all overlapping. So I would say, what's the effect? It seems like exercise does not have a statistically significant effect on blood plasma prolactin. Predict how your graph will look if we change X or Y and justify. This is what we kind of said earlier. Maybe I've got an enzyme graph that's going like this, and with pH down here, then they might say, what happens if you increase the pH? This is like pH, 4, 5, 6, 7. What if you increase the pH to 14? Well, then I might think that it could go up for a while, but it might denature, and or the number would flatten out or decrease. And then why justification would be, well, you'd have to explain denaturing. So predict what would happen. My graph would go up, down, stay the same, whatever, and then justify, oh, here's why. What's the significance of group A? So they might come in and let's say that I test green versus yellow, and then this one has water. They might say, all right, what's the significance of this group? And you're like, oh, that's my control. It's a point of comparison. And they might explain like why that's necessary. Have you explain why that's necessary? Like, why do I need a control? Well, this is a negative control. And it's a point of comparison to see if these guys actually make a difference. If my control and my experimental groups, these are my experimental groups, if my controls and my experimentals are similar enough, then we don't think the independent variable of putting in these active ingredients seems to matter. Propose a valid control. Sometimes they'll give you this, and they don't give you this. And so now you have to say what a valid control would be. And don't just say, I give them water. Say, if these are being applied with wipes, I'd say, I would get a similar type of wipe that was sterile, had no added chemicals, and I would add water to it, and then I'd wipe it the same way or something. So go out of your way to try to explain it a little bit. It might not be necessary, but a control group for this, a negative control would be like water, for example. Like I said, I will down in the description link a few practice problems related to this, and I think this will help you a lot on question number 1. All right, thank you.